Thank you, Becky. Thanks everyone, and welcome to our both speakers and all of you. So our first speaker will be Paul Freemont, and then we'll move on to Mike Hammerling. So starting with Paul, Paul is a professor at Imperial College of London. He's one of the best, most accomplished British synthetic biologists, hardcore synthetic biologists. He's a co-founder of the Imperial College Centre for Synthetic Biology and Innovation and director of the National UK Innovation and Knowledge Centre for Synthetic Biology. He's a director of the London Biofoundry, and he helped establish the Global Biofoundry Alliance, among other of his directorships and accomplishments. His research group pioneered the use of cell-free extracts, cell-free systems for synthetic biology in general, for prototyping, and for biosensor applications. And I won't take any more of your time, Paul. It's all yours. You have to unmute yourself.
Yes, thank you so much, Kate, for inviting me to this first seminar series, and also to Becky for organizing it. So I'll just share my screen and we can crack on. Firstly, I wish everyone is well and coping with everything that's going on in the world. Incredibly, we're very united in all facing the same challenges. So it's a really strange period, but I'm really happy to be able to. So I think I'll just move straight into my first slide.
Okay. I'm going to stop you for a second there, Paul. We're seeing your Zoom screen. Okay, that looks better.
Is that better?
Yes.
Yeah. Sorry about that. I'm out in the countryside. I live out in the countryside miles away from London, so we don't have many internet connections, strangely. So anyway, synthetic biology considers DNA in part as a programmable language in some ways, and there are some significant challenges. So I just want to remind people about the challenges, and then I will move on to the talks. The thing about engineering and reprogramming biological systems at the genetic level, it's very important to remember that there is context dependency in biological systems, and of course the evolution, adaptation, and natural selection are key aspects of every living system. And then you have this phenomenon of non-predictive random behavior, and then this really amazing thing of self-assembly, which then leads to emergent properties. You have a lot of nonlinear dynamical processes and steady state, and of course you have multi-scale interactions between the electrons flowing down a particular electron transfer protein system through to whole organisms. So there's a huge scale issue, and so cells, I suppose, on the right-hand side there, are more like these kind of wraps, and they're not circuit boards, as everyone I'm sure appreciates. So, I mean, one of the things that SynBio has kind of established to overcome this is to try and define a systematic design framework. And of course, as you all know, this is very much built around this design, build, test, learn kind of cycle, which then of course leads on to the aspects of biofoundries and automation and all sorts of systematic design and metrology and measurements. And I think this is where I think synthetic biology is hugely impactful and making enormous progress in many other areas as well, but particularly around the biofoundry development. So for cell-free in vitro transcription and translation systems, they offer new opportunities for these kind of synthetic biology workflows. And so if you look at a cell-free system, I think most of you realize I'm talking about cell lysate systems here rather than the pure system. So we're looking at cells that have been grown and lysed, and they're essentially the machinery and biochemistry harvested into a tube. And then we have the wonderful ability of being able to add in genetic constructs, either linear pieces of DNA or whole constructs, to allow you to essentially execute, if you like, the DNA code within a cell-free lysate. And the lysate has all the machinery to be able to translate that code, to transcribe that code, and to translate that code, and produce outcomes that you can measure. And on this slide is just an illustration of some of the circuitry that you could think about. Also on the right-hand side is the wonderful ability to measure both an output, for this is GFP, for example, and also to measure messenger RNA production by using sort of clever designs and various dyes that bind onto aptamers at parts of your messenger RNA molecule. So you can have real-time measurements, which I think is very interesting. Then the other thing to point out is that Cellfree systems are not new. They've been around a long, long time. I won't go through this in too much detail, but needless to say, in the 1960s, they were really important for proving the genetic code. A tour de force in 2001 by our Japanese colleagues that created essentially a whole reconstituted transcription translation system, which proves to be a fantastic achievement and also incredibly useful for people wanting to study the very basic fundamental aspects of transcription and translation and try and reconstitute that. And then, of course, we started in the journey of synthetic biology, late 90s, early 2000s. And of course, in the US primarily, there were some real pioneers in cell-free systems, James Schwartz, Vincent Wiro, Michael Jewett, for example. And then in Europe, Sebastian Merkel and Sven Panker and Richard Murray at Caltech and our lab to some extent have contributed to the realization that cell-free systems can offer an enormous opportunity in a synthetic biology design context. And I think also many labs, including Mike's and other labs, have and Keith Pardee now and other groups and Lux's lab in Northwest have really grabbed hold of this and taken the application space into many different spaces. And I think, that is really exciting. So we cell-free systems have been used a lot for sort of optimizing protein synthesis. But when you start thinking about new vaccines, distributed biomanufacturing, low-cost sensors, new chemical synthesis, even education and outreach, with the opportunities of teaching young colleagues and school children how to engineer living systems, and then of course natural product biosynthesis, and then a prototype in gene expression. So those are named but a few, but I think to me that's an extraordinary array of different opportunities and excitement in the cell-free lysate field. So prototyping parts and design. So this is the concept of cell-free lysate prototyping. So the idea here is that you would have a series of host cells that you would be interested in engineering. You would then make cell-free lysates from those host cells and characterize them and make sure they're functioning. And then you would have the opportunity of designing or even creating libraries of new parts for those particular host cells, particularly non-model organisms, which I'll tell you about. And then the opportunity of doing computational modeling and looking at designs within a cell-free lysate context gives you this kind of mini, if you like, design, build, test, learn cycle, but within a lysate context, which allows very much high throughput, which you'll see in a minute. Of course, the assumptions of all of this is that the things I measure in vitro have some representation or some reality to in vivo measurements, and that's a key part of this, at least for the prototyping. And a little while ago, an ex-PhD student of mine, Jim Chappell, came up with the idea of testing this hypothesis, and we did that and found actually surprisingly that you could get, for simple regulatory elements like promoters or ribosome binding sequences, pretty good agreement between in vivo and in vitro measurements. And this was relative agreements, not absolute agreements, but still strikingly interesting at that time. And it made us think that, well, maybe you could start using cell-free systems as prototyping and measuring things that could have some relevance for in vivo. And then, of course, for cell-free lysates, you have the ability to do very small volume reactions. You also have the ability to do high throughput. So we have developed a number of workflows using an acoustic liquid handling robot. And that allows, fast, scalable, robust cell-free assays. And, you can go right up to 384 well screens. We've done that. You can use 2 microliters as an assay volume. And it leads into the concept of if you've got 50 mils of cell lysate shown here, you've got a lot of reactions and assays that you can explore, which means that in a design space or in a part space, that really opens up opportunities for you. And so if we think about prototyping parts and designs, we focus a little bit on non-model organisms. And I think the reasons for that, from my perspective, is that, they're very interesting. They tend to not be studied very much. They tend to be very little genetic elements available for engineering these organisms. And they often tend to have very interesting properties or phenotypes. So just on that slide, you can see the extracts contain native regulatory machinery. Maybe they have specific cofactors, maybe some native chaperones for folding. New regulatory parts can be prototyped quickly. And of course, you can start thinking about unusual biochemical synthesis, maybe some natural product discovery in particular organisms, which I'll talk about. So our first one was Bacillus subtilis. We published this, so I'm not going to mention anything to do that, but it's an interesting organism, industrially relevant, and we quickly showed, in that slide, 3 measurements, both in vitro and then in vivo plate reader and in vivo flow cytometry, and that shows the agreements between a synthetic promoter library that we were working on at the time. So again, reinforcing the concept of in vivo and in vitro having direct relevance. Then we moved to Bacillus megaterium. Again, we published this in 2018. It's an interesting organism, again, industrially relevant, absolutely very little genetic tools available. Quite an unusual organism because that's the organism which is, as the name would suggest, it's very, very large. That's E. coli in orange there. And it has got a secretion apparatus. And a lot of people are interested in using this maybe as an industrial chassis. But as I said, very few parts available. So we developed a workflow for this particular organism. This is one example, just showing how quickly we could prototype the native promoters that we pieced that up from the Bacillus megaterium genome, which covered all of the native glycolysis and gluconeogenesis promoters, and measured them in vitro. And that's shown down here, where you've got messenger RNA, as shown on the left, and then protein production as a GFP reporter on the right. And then a tour de force by our collaborators actually allowed to do some in vivo in vitro measurements, and again, getting reasonably good relationships between that. But it was an exercise in showing how quickly one could get parts for this organism, and this organism has so few parts, and none of these have ever been characterized before. And another example of that is this part library of a ribosome binding sequence library we made very rapidly using the Bacillus megaterium system. It also allowed us to do a lot of modeling, but because of the time constraints, I won't be able to discuss that, but please go and have a look at the paper and you'll find all that information in there. The next organism we moved to was Streptomyces, particularly Streptomyces venezuela. Now, of course, Streptomyces or actinomycetes have an extraordinary potential of natural products, and people have been working for many, many, many years. Many of the antibiotics, some potential therapeutic cancer drugs, have been developed from actinomycetes, this particular bunch of bacterium. And I think there is an excitement there about exploring the Cellfree system, not only for prototyping parts, but also for maybe some natural product discovery. So Simon Moore in my lab, who's now got his own group at the University of Kent, pioneered this work. Sorry, on the previous slide, Simon and James, you've seen the names of the people. Sorry, I haven't mentioned them, but I'll come around to it at the end. The people in my lab were doing this. But so Simon was pioneering the Streptomyces and managed to get a very nice expression system going, and also did some characterization and prototyping. And one example, this is the expression system here. And you can see it took a little while to get a good continuous, semi-continuous expression yield of proteins using the Streptomyces system. But that was good enough for us to do what we wanted to do, which was to go on and look at some synthetic promoters. And we did this and some ribosome binding sequences. So we took out of the literature a whole bunch of synthetic Streptomyces promoters, and we plugged them into the Cellfree system and characterized them. And on the right there is the correlations between in vitro Cellfree and the cellular in vivo data as published in this PNAS paper by Lijiang Zhang in Shanghai. And so again, illustrating the possibilities of rapidly prototyping parts for this particular organism. We then most recently moved to mammalian systems. I'll come back to Streptomyces later on. We moved to mammalian cell lysates. And this is a paper that we just published from Margarita Kovnitsky and Kiva Canavan. Margarita's now graduated, a PhD student. And what Margarita did was she took a whole bunch of constructs as shown there. So a T7 promoter library, an IRES library. She explored this DCAS9 transcriptional repression possibility using guide RNA directed repression. And then she also looked at a small archaeal RNA binding protein, L7AE, and looked at direct repression of transcription and translation. And then she developed a workflow. So this has all been published, so I'm not going to go into any details, just one little slide to show how one can really quickly do a bunch of T7 promoters. And of course, the thing to remember about mammalian systems is that you're working on a cytoplasmic lysate. Of course, the nucleus is lost in the preparation. And one of the things we're working on, and other people are trying to work on, is to have a completely linked up transcription translation assay where you can start exploring endogenous promoters for mammalian systems. But that is quite biochemically challenging. So a lot of transient transfection experiments often use T7 promoters or iReses. So some of these experimental results, at least for the prototyping, is still quite relevant for people wanting to explore expression in mammalian cells. Building biosynthetic pathways. So we set ourselves the task of maybe trying to recapitulate the synthesis of oxytetracycline, which is probably a little bit too ambitious at this point, but it was an interesting idea. And this is the biosynthetic pathway of oxytetracycline, and you can see it's a series of complex enzymatic transformations with some interesting cofactors that are unusual and relatively rare, particularly coenzyme F420. And we set about to express all of the enzymes in this pathway in the Streptomyces cell-free extract system. So this is looking to see if we can actually make the enzymes directly. And again, this is the work of Simon Moore and other colleagues that Simon was supervising at the time. And you can see from this slide that we were able to make quite a lot of the proteins. So on the top there, you'll see all of the proteins being made in quite good yields. You can see on the bottom right here an SGS polyacrylamide gel with bands illustrated. So we're getting good expression yields. They were all soluble. We tested 19 proteins so far. We've got this non-ribosomal peptide synthetase also expressed. And we're now just beginning to explore whether we can get any catalytic steps going between these particular enzymes. But I think as a proof of principle, it looks promising that we could express a lot of these excuse me, enzymes in Streptomyces, and 1 or 2 were actually insoluble in E. coli. We also did the melanin biosynthesis using the Streptomyces cell-free extract. And so this is, Simon took a 3.6 kb little mini operon here and explored the chemistry of that and was able to show us adding in tyrosinase you could get melanin synthesized, which was great. And then also he took this 6.1 kb operon with a bunch of enzymes, which is involved in heme biosynthesis in Streptomyces, and again showed you could express these enzymes and get biosynthesis of some of these heme-like molecules, which we could analyze by the LCMS, and this is work he's writing up at the moment. Now, I want to spend more time on this part of the project, which is making novel xenobiotics. So we've been focusing quite a lot on violacin, and we're interested in violacin because it's a natural product. It's produced by a very well characterized pathway. It has a broad spectrum of biological activity, as you see on this slide, antibacterial, antifungal, whole series of interesting biological activities. It has been shown to disrupt Staphylococcus aureus. There is an analog, oxyviolacin, which has some improved biological activities, but it's clearly an interesting molecule to study. And one of the things we wanted to try and do is to see if we could somehow develop a strategy to create derivatives or chemical derivatives of violacin or deoxyviolacin. So this is the work of Hong En Lei, who was an ex-PhD student who's now down working in the University of Victoria in New Zealand. And this is a collaboration with Rebecca Goss in St Andrews. And the idea here was that could we combine both in vivo biosynthesis, so build a cell that produces the violacin molecule itself, and then somehow could we substrate feed into that some tryptophan analogs that would allow the derivatives or the chemical analogs, if you like, to be taken through the whole biosynthetic process and end up resulting in violacin molecules that would have these chemical derivatives. And so this is a sort of summary of that. So the idea was that you'd have E. coli expressing the violacin pathway, you would put in these synthetic tryptophan analogs, you would then produce a series of, hopefully, of violacin derivatives, One derivative in particular, which we would be very interested in making, was the bromoviolation derivative, because then that would allow you to that would allow you to catalyze further chemistry using synthetic organic chemistry methods and approaches to try and even further derivatize any bromoviolation scaffold as shown here. And the whole point of this was to try and generate a series of violation analogs that could then be driven through into a biological assay and see if we can create, by this combined in vivo in vitro method, interesting analogs with improved biological activities. So here's the violation pathway, via A, by B, by E, by C, by D. It starts, as I said, with tryptophan, and here on the bottom is the tryptophan analogs that we tested. So for fluoro, chloro, hydroxy, fluoromethyl, and 7-methyl, and that just shows you the distribution of where those different side groups are. And the idea was to feed that into the pathway and then have catalysis move its way all the way along, and then hopefully create a whole series of interesting violation products. First point in this was that as we're feeding things into this first enzymatic step using BioA, it's really important to see if that enzyme would actually work. So Hung-In actually did the crystal structure of BioA, and this is the structure he solved. And you can see here that there are the tryptophan and the FAD cofactors shown in the active site of the enzyme. And there are a couple of key distances, and particularly this distances between the C alpha proton right through to this tryptophan amide. This is a key distance. And essentially this first reaction is essentially a proton abstraction reaction. He then soaked in all of the tryptophan analogs shown here, and surprisingly, they all bound in slightly different orientations. But importantly, what we did observe was that the contact distance between the proton on the carbon alpha of the tryptophan analog, the distance was very similar to the wild type, meaning that the chemistry, at least from this analysis, could proceed. And here is the biochemical activity assays of that first step, showing that you've got tryptophan, 5-methyl, 6-fluoro, 7-methyl, and all showing reasonably similar Michaelis-Menten kinetics, not identical, but certainly showing catalysis, at least in vitro purified enzyme. And at least that gave us confidence that we could get our analogs into the first step and at least get them into this interesting 2-IPA imine derivative, in the hope that then that would condense and then lead to the violation analogs. And to cut a long story short, this is the essentially. Also, we did a lot of metabolic engineering as well, where we improved the violation yields, which I won't really talk about. But to cut a long story short, we ended up producing a very large diversity of violation analogs. You really can't see all of the different diversity here, but you can clearly see from of the R groups that how many different R group combinations you could actually manufacture or make using this system. And so when we analyzed all these by LCMS, and we did some sort of semi-quantitative analysis of yields, that's what's shown here. So we were able to detect 62 violation analogs tracks expressing the pathway. We use combinations of analogs. So here's 4-fluoro tryptophan. This is 6-fluoro with 5-hydroxy, et cetera, and these pie charts just indicate violation, which is the normal violation, because you have endogenous violation being produced as well, deoxyviolation in red. And so for example, in this particular result, we were unable to get any derivatization for that particular extract. But then all the others, they show different variations of extracts compared to deoxy and oxy. And of course, you would have deoxy and oxy for each tryptophan analog as well. So that's a sort of summary, if you like. And what was interesting then, we were able to make a chloro analog, but this time we made it in vivo. So we could replace a couple of enzymes and get a tryptophan into a chloro and then take that through to the deoxy version. But more importantly, we were able to make the bromo. Now, that led us into a whole interesting space, because then you could think about applying this type of interesting synthetic organic chemistry. This is a Suzuki-Miura coupling, where you can conjugate two molecules via a palladium catalyst and create very interesting subgroups. Now this is where the Cellfree comes in. So here we've got our living cell producing our 5-bromo-L-tryptophan. We then essentially harvest those cells and make cell extracts from them containing that product. And then surprisingly, we are able to carry out the actually synthetic organic chemistry within the context of the cell extract. And this is the work of Alan Obled and Rebecca Goss's group. And he was able to create conditions to allow the production of this first toyol, a violation derivative, which was fantastic because that then led us into a whole series of 15-aryl boronic acids that could be used to create this whole beautiful diversity of violation analogs. And we're analyzing some of the, Alan is analyzing some of these right now, and we're very excited by these results. Now, of course, this all is exciting, but what does it mean? Does it mean anything? And so the last few minutes of my talk, I just wanted to show where we'd got to on the biological testing of the data. And basically, we picked up a really great collaboration with Jake Baum's group. He has a screening and growth inhibition assay for Plasmodium falciparum. And we just plugged our stuff into him. And this is the assay. I won't go through it, I haven't got time, but it's a very well-defined assay. And what he's looking at is it's a parasitic growth inhibition assay. So he's looking at the inhibition of the Plasmodium parasitic growth within red blood cells. And he also found that violacin actually has got some activity in this particular assay. So shown here, you can see this is the violacin from Sigma that you can buy, and this is the growth inhibition. So the percentage growth inhibition on this axis, and then the drug concentration here, so you can get a nice sort of KD or IC50 if you want. And then over here is the violacin that we synthesized, and down here is the chloroviolacin, and of course we did a lot of experiments on all of these. And this is the summary slide, my last slide. And this shows you, this is the percentage inhibition, 50% as an arbitrary number. This is the violacin from Sigma. This is the violacin we made biosynthetically. And then across this whole spectrum are all of these derivatives, 6-fluoro, 4-fluoro, 6-methyl, whatever. What was really interesting to us is that we were getting some really striking inhibition, comparative and relative inhibitions from different derivatives, which actually was really extraordinary, really. So we're now exploring some of these in great detail and trying to explore further. And of course, the next series of analogs that Alan is making will plug into this assay as well. So summary, I've ran through a whole lot of stuff there very quickly. I hope I haven't been too quick, but the cell-free protein synthesis assay is used for a variety of applications. I focus mainly on some of the prototyping for non-model organisms and some of the small biosynthetic pathways that we're exploring, and then spend a bit of time showing this really interesting combination of using in vivo and cell-free extracts to do synthetic organic chemistry and create new chemical analogs and chemical scaffolds, which could be useful for any drug discovery or any sort of biological activity measurements you're interested in showing. That is it. The work I've described, Simon Moore, who's now the PI, set in his own group in the University of Kent, did a lot of work, as I described, on Streptomyces and the Bacillus megaterium. Hung and Lei, now at Victoria University of Wellington, New Zealand, did a massive amount of work on the violacin project. James MacDonald, who did work on the Bacillus megaterium project and did a lot of mathematical modeling, which unfortunately I didn't have time to talk about. Kirsten Jensen, Alexander Webb, and Richard Kelwick have all been instrumental in aspects of the project. Margarita and Kiva I also described. So thank you very much, and I hope I wasn't too much over the allotted time.